Morning all.
Good morning, sir.
Good morning.
Ute, Papa Ute, Papa Ute, Papa Ute, 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 Papa Ute, Ute, Papa Ute, Papa Ute.
Please, you are live on the intellectual class.
Your Papa Uche.
In case you don't know.
Nobody know me bye.
Obonie.
Meiosis
Hello? Hello? Can you unmute and respond?
Good morning, sir. Good morning, sir.
Sir.
Good morning, sir.
Good morning, sir.
Good morning, sir. Good morning,
There
sir.
was a serious technical
Good morning, sir.
which may affect our live streaming in YouTube. Can we just continue on Zoom like this? I think there's still space for more people to join.
Yes, sir.
Yes, sir.
It's like the YouTube is not really working. That has been causing the problem.
Yes, sir. It's working, sir. The YouTube is working, sir.
Okay.
Okay.
Oh, you can hear me through the YouTube as well.
I'm coming, sir. It's working, sir.
Can you still
It's okay, sir.
listen to me? The YouTube is okay, right?
Okay, sir. Okay.
Yes, Papa.
Yes, sir. It's okay. I can hear you through the YouTube, sir.
Okay.
This one is it.
Can you see what I'm sharing now? Can you see what I'm sharing?
Yes, sir. We can see your slide, sir.
Yes, sir.
Yes, sir. We can see the screen.
Yes, sir. We can see the screen.
You can see my
Yes, sir. The slide is showing on YouTube now. The slide is
Can
showing on
you
YouTube
see?
now.
Okay.
Yes, I can
Those
see it
on
on YouTube, sir.
Zoom, can
On
you
YouTube, I can see it on YouTube, sir.
on
On
Zoom,
YouTube, I
on
can see it,
Zoom?
sir. On YouTube. On Zoom, we can see it also.
Those on Zoom, can they too see it?
Yes, sir. We can see
Yes,
it on
sir.
the
We can
Zoom
For
see
too.
it.
those on the Zoom?
Yes, sir. We can see.
Yes,
Yes,
we
we
have
can
seen
see
it,
it,
sir.
sir.
Okay. All right. So our time has passed, but since you don't have any other class now, can we continue?
Yes, sir.
Yes, sir. That's fine.
No.
All right.
No.
Okay. Once again, good morning. My name is, as shown on the slide, and the topic that I'm going to take you, okay, is on cellular reproduction and heredity. So if you can hear me, just mute yourself so that you don't disturb the flow. So my name is Dr. M. Shifao, and I'll be taking you on cellular reproduction and heredity. To start with, under cellular reproduction, we have what is called cell cycle. This cell cycle is another sequence of events that occur in a cell in preparation for cell division. In other words, before a cell division will take place, there are sequences of events that will precede cell division cycle. And you know, when something is in a cycle, that means it's something that occurs every now and then. Start, complete it, start again. So it's a recurrent event. There are 4 stages in the cell cycle. We have the G1 or gap 1. We have the S stage, and we have the G2. So also we have the M stage. So G1, S, G2, and M. The G simply means gap. That is a gap between a stage and the other one. So what happens in that gap? What happens there is the cell increases in size. The cell will increase in size. In synthesis, of course, from the name, that's the S stage. What happens there is the cell will duplicate, will copy its DNA. There is duplication there and synthesis of other materials. Then in G2, the cell will prepare to divide. So after increase in size at G1 and synthesis of materials that will be needed, so also duplication of DNA, then at G2, the cell will now prepare to divide. But G2, but also try to do what is called in production industry, quality check, quality assurance before they divide. So at G2, while the cell is preparing to divide, it will cross-check all the synthesis that took place at S stage. So if there is any error in duplication or in synthesis, the errors are corrected at G2. So the error will stage, that is the mitosis, the mitotic stage. If perchance the error is allowed to go, then it continues and leads to cell aberrations and leads to cellular disorder that will take place. But the cell cycle is structured in such a way that those errors will not be allowed to pass through. They will be corrected. There will be a kind of quality check before they go into division, which is the M stage. Also, in a cell cycle, as I said, is divided into 4. The stages G1, S, and G2, they make up the interphase, which accounts for the span between cell divisions. Like I told you, a cell division will continue to occur. So it takes place like in a cycle. So a cell cycle is broadly divided into 2. We have the interphase and we have the other phase. That is the division stage, G2, which I have told you what happens at these stages, the G1, the S, and the G2. So, on the basis of some stimulatory and inhibitory messages that goes into a cell, then the cell is able to decide whether it should enter the cell cycle and divide or not. So if there are inhibitory messages that the cell is receiving, then the cell may not go into division. But if there are stimulatory messages, then it prompts a cell to go into division. So these are balances and checks for a cell to proceed. That's why at a certain stage in the lifespan of an organism, the cell division, the level at which a cell divides reduces. Unlike when the cell is still young, when the individual is still at a young age, that there is active cell division. So when a cell is aging, okay, the rate at which the cell divides also reduces. So this is the cycle. So when you look at the cycle, you see this is the one inner. You see that you have the interface that is in purple color, and you have a certain say which is pink in color. That is the the broad division that I said a cell cycle will be. That's into two. So you have the interphase stage and we have the mitotic phase. So when you look at the interphase, you see that that is where G1 occurs, S, and G2. And like I've told you, at G1, cellular contents, excluding the chromosome, are duplicated. Okay, you will see that at G1, duplication of chromosome does not take place. All other things in the cell, okay, will be duplicated. Like I said, the cell grows at G1. And when you get to S stage, that's where you have the chromosome duplicated. And then at G2, there is a kind of cross-checking what has been produced at S stage so that there will be no error of mitosis. They will not be allowed to go into actual cell division. So they will be corrected at G2. Okay. So there is a stage, but which is not known. That's the G. You can see that it is here outside the cycle. Your cell after division can proceed to G0 if there are messages, inhibitory messages. Like I said, if there are inhibitory messages that a cell receives not to continue into another cycle, then there is G0. That is where a cell is said to be resting. Okay, so with this, get this to know, the cell does not rest at a stage of so many activities. Like you can see at G1, at S, and at G2. So a cell only is outside the cycle. So if a cell receives such a message to hold on before continue with another division, Then the cell enters into G naught. So G naught is a phase rest and not interphase again, right? So these are some of the activities and the duration of each of them. So, like I said, interphase is a phase with highest metabolism. So, and you know, metabolism has to do with anabolism and catabolism, where mitochondria have the highest activity. So when you have highest activity, do you rest? So correct that notion that you have from your secondary school biology that at interphase is a resting state. Interphase is not a resting state. Okay? There are other activities apart from chromosome duplication. So protein synthesis and growth. The cell grow and synthesize other materials. And you can see that the duration G1 takes is the highest, 10 hours for all those things to be complete. It will stay there for 10 hours, okay? And then also prepare the cell for replication. And also at G1, okay, there are differentiation where only selected genes are used to perform different functions in each cell. So there will be differentiation at G1 as well. Then at S, purely is DNA replication. DNA replication, majorly DNA replication. So that one takes like 9 hours. Okay. At G2, I told you G2 is a short gap because what happens there is to cross-check what has been produced at G1 at S. Okay, there is a kind of quality check before they come to the next phase. So it's a short gap, long, and you have about 4 hours there. Okay, so the G naught, like I said, is a resting phase where some cells can rest unless they equally for them to continue with the cell division, cycle. And you have example of nerve cells. Nerve cells, they don't divide every now and then. Okay, after some division, they stop. Don't go ahead again. Then we now have the M phase. M phase is the other. Okay, so interphase is G1, S, G2, and G naught. Then the other one is M phase. This M phase is the phase of cell division. And we all know that there are two phases or two ways by which a cell can divide. The two types start with M. So we have the mitosis and we have the meiosis. So you will see that it's only mitosis that is written here because mitosis continues in the cycle. But meiosis has some unique features. So when we get there, we'll see. So we are going to take the M phase now, starting with mitosis. phase, starting with mitosis. So mitosis is a process of producing two diploid daughter cells. Please be vigilant about some of the words that are being. You should take note of them. Two diploid daughter cells. Two cells. Okay, but the two cells are what? Diploid. They are 2N. 2N. And with the same DNA. The same DNA. And this is, I mean, their chromosomes. So it's like a kind of cloning. This is the duplication. Duplication of chromosome took place in the S phase of interphase. But that's the chromosome, not the cell. So on getting to mitosis, the cell will now divide so that the duplicated chromosome will now be the same in the two daughter cells. Again, mitosis is a control process, okay, as against cancer. You know, cancers too, you have the cell dividing. Cancer results from uncontrolled mitosis. So if a cell continues to divide without control, it leads to cancerous cells. Mitosis involves two processes. We have the karyokinesis and we have the cytokinesis. The karyokinesis is the division of the nucleus, which comes first before cytokinesis. Cyto means cell, before the division of the cytoplasm of the cell into two, each containing one of the nuclei. That must have been produced or divided at karyokinesis. Mitosis also occurs in 4 stages. 4 stages, which are prophase and telophase. In our secondary school biology, we are told that interphase is part of mitosis. Mitosis and telophase. However, so the 2 phases of mitosis and the occurrence that take place in each of the phases. Now, when you look at this, you will see that this portion was brought out from the cell cycle. Remember the cell cycle we talked about previously. And phase, M phase, which is the mitotic phase of the cell cycle. When you look at before prophase, interphase, okay? And after telophase, you see cytokinesis, which is the cell division, the cytoplasm divides. So after prophase, metaphase, anaphase, and telophase must have taken place. In some textbooks, however, you can see another stage being included in mitosis to make it 5. That is pro-metaphase. Pro-metaphase is a phase which is not in all cells. It's a phase between prophase and metaphase. That's why it is given that name. Pro, meta, that is, prophase is up here. Metaphase is that, so it's in between. And what takes place there is also given here. Now, briefly, the 4 phases of mitosis, okay, that are the major events in prophase. Okay, number 1, we have breakdown of nuclear membrane. The nuclear membrane surrounding the chromosome will break down. Then we have the spindle fibers appearing. Okay, then the chromosomes condense. Chromosome that before prophase, they were not distinct. They were just loosely coiled. When you look at G2, you see the chromosome there that are not really distinct. But at prophase, the chromosomes are now distinct because they condense. You know, when something condenses, you have to recognize. So the major events at prophase are these 3: breakdown of nuclear membrane, spindle fibers appear, chromosomes condense. These are, mind you, but these are the major ones, at least for our level here. Then pro-metaphase. At pro-metaphase, okay, that's why I said is a phase between prophase and metaphase. And you can see what started from prophase continues at pro-metaphase, like spindle fibers that appear in prophase. Okay, at pro-metaphase, they did not just appear, they are attached to the chromosome. The spindle fibers are attached to the chromosome, and again, we have further condensation of chromosome. Chromosome condensation continues. They condense more so that they become fatter, okay, than before, and become more recognizable. So at metaphase, at the occurrence, major occurrence at metaphase is the alignment of all the chromosomes. You can see them individually, they align. They didn't pair up, they align individually. So that's the major occurrence at metaphase. I mean, at metaphase, alignment of chromosomes at the equatorial region of the cell. That is at the middle part of the cell. They align there. Then at anaphase, okay, what happened? Remember, a chromosome is joined by centromere. There are 2 chromatids in a chromosome, and these 2 chromatids are joined at they are joined by the centromere. So when they align at metaphase, on getting to anaphase, what joins the 2 chromatids together will divide, so that you now have the chromatids being separated. The chromatids are separated because what joins them together would have been cut or divided. So at anaphase, we see the movement of each of these chromatids towards the opposite pole. So at anaphase, the major events there are division of the centromere to separate the chromatids. Then we now see the sister chromatids moving to opposite pole. So the sister chromatids here, two chromosomes are illustrated, red and blue. And in each one, you see two chromatids. So when they have been separated by the division of the centromere, each chromatid, okay, each chromatid of a chromosome moves to the opposite pole. So telophase, simply put, what happens at telophase are just the opposite of the major event at prophase. You know, we started the cycle with prophase, with all those three major events. However, at telophase, the processes at prophase are reversed. That is the opposite. For instance, look at telophase here. We now see nuclear membrane forming again, reformed. But at prophase, we said nuclear membrane breaks down. Okay, they break down, they disappear. But here at telophase, they are now forming back. Right? Now, at telophase, chromosome decondense. They decondense. That means they are now becoming longer and no longer fatter. Because when they condense, they become fatter. When they decondense, then they become, you know, longer and thinner. They are no longer fat again. Please, can Simon Sure mute his or her video? I don't know why you are moving about. Close your video as well, or I send you out of the play of this class. So I've been watching you, you are just moving up and down. Simon Sure, that's the person I'm talking to. So that's that at telophase. The third one is the spindle fiber. The spindle fiber disappears. Whereas at prophase, we said the spindle fiber appear. So that's why I said the three major at telophase. So as you all can see what happened there. And again, at telophase, you see the cell a different shape. Rather than circular in nature, is now assuming a kind of figure 8, okay, in preparation for the division of the of the cell to form two daughter cells. So that's what happened at cytokinesis. Cytokinesis, you now see the two daughter cells. Okay, because the cytoplasm has divided, it now give us two daughter cells. So apart from the division of the cell at cytokinesis, we see that parent cell becomes two daughter cells, right? And with identical genetic information. When you look at the content of each cell, you see the red and the blue, which are chromatids. Okay, the chromatids from the chromosome before they separated. So we now have a representative of each chromosome in each cell. And that's what makes them to be identical or similar. So they have identical genetic information based on So that is summary of phases of mitosis. What I have next is what I have just showed you before. In M phase, in interphase, the longest phase is the G1. Okay. So also in prophase of mitosis, the first and longest of mitosis is the prophase, and during this, chromatin chromosome feasible nuclear. Okay, so all these are said. Oh, then the spindle fibers form in the cytoplasm. That's at prophase. Then metaphase, spindle fiber. Okay, pull sister chromatids to line up at the equator or center of the cell. They all line up. Okay, and if there is error in the way the spindle fibers attach to chromatid, it can lead to uneven or genetic bias. That's at metaphase.
Sir, we can hear you, sir.
Sir, we can hear you.
Can you hear me now? Okay, I said
Yes, sir.
at interphase,
Yes, sir.
spindle fiber begin to short. You can't see hear me. Who cannot hear me?
We can hear you now, sir. Yes,
I can
sir.
hear you, sir.
Okay. Yeah, I just move a slide from there. So anaphase, spindle fibers begin to shorten, and by shortening of the spindle fiber, they move the central. They move the chromatin towards the opposite pole. And our telophase, like I said, telophase is the phase, because what happens at prophase happens at prophase. But at telophase appear. Spindle fibers appear at prophase, but at telophase, the chromosomes are condensed at prophase, but at telophase, they decondense, that is, they are uncoil, so that they now become thinner and longer. no longer becoming fatter. Then cytokinesis, which is the final stage of the division. Mind this, cytokinesis is not part of M phase, because we have seen a situation where after telophase, it will not lead into two separate daughter cells. They continue joining together and continue. We see this especially in embryonic development, where we have the first diffusion cleavage leading to two cells, leading to three cells, but all still join together, not leading to separation. So in many cells as well, cytokinesis leads to two separate daughter cells with identical nuclei. Two separate daughter cells with identical nuclei. So the process of cytokinesis in plants is different from how it happens in animal. But all the same, all the same cytokinesis leads to two separate daughter cells. So in animal cell and in plant cell, this is how cytokinesis takes place. The one on the left side is the animal cell, and the other one on the right side is the plant cell. Which we now put like a depression on both sides of the cell. So look at this. I told you the cell will now change shape to something like a figure 8, so that the depression continue until there is a touch between the two, and then there will be separation. So cytokinesis in animal cell occurs by formation of cleavage furrow. In plant cell, it is by formation of cell plates. So the cell plate will cut across the cell. It's not a division. It's not a depression, unlike in animal cell. So once the cell plate cut across, it will now create like a middle wall or middle lamella, and then lead to the two separate cells. And you know why it has to go through that route is because a plant cell contains cell wall, which makes it difficult to undergo the way cytokinesis happens in animals. So cleavage furrow cannot form in plant cell because of the cell wall. It will not allow it. So in plant cell, cytokinesis takes place by the formation of cell plates, which cut across the cell and divide it into two. So that's that for the division, mitosis. However, there are some significance or importance of mitosis. Mitosis is important for the maintenance of the chromosomal set. You know, a cell contains 2 ends of chromosome. That's the diploid number. So if that cell is divided into 2, each the 2 and diploid. So mitosis chromosome set. So in each succeeding generation is always 2n. So each cell form receives chromosomes that are alike in composition and different cells. The chromosomes that are formed in each daughter cell are alike. Why are they alike? Remember, each chromosome will separate into their two chromatids. So a chromatid will go into a cell, the other will go into another cell. So why would they not be alike? So they will still be alike because each chromatid, okay, of chromosome are represented in the daughter cell produced. Then the number, the 2n is also constant. If 2n is 4 in the parent, you can be assured that 2n will be 4 in the daughter cells produced. Another significance or importance of mitosis is in growth. If there is no mitosis, how are we going to grow to the age that we have now? Each one of us was born. We are very small. They are so small that they are hardly noticed. Even when they are on bed, they say, hey, don't sit on the baby. They are very small. Now, with time, that baby that they are saying people should be careful not to sit on. He's not a big boy or a big girl now. So it has a reason. This, okay, is responsible for repairing of some damage, okay. Mitosis will repair them so that the normal life activity of the organism will continue. And there are some cells that will die, cell die, and new ones are produced. So if there is no replacement of dead cells, many people will not grow up to now, up to the age they are now. If cells in them continue dying without replacement, the individual is dead. So as some cells are dying, there are replacements that we have. Example, red blood cell. Red blood cell has a lifespan, okay, and once it attains the lifespan, it dies. And when there is no replacement, the individual is equally close to death. Then we have skin, or you peel off some skins. Those are dead ones. And you have a replacement beneath that one. So if there's no provision for that, I don't think people can survive. Then asexual reproduction. Asexual reproduction, this is common in unicellular organisms. Unicellular organisms that don't have sexual organ. How do they reproduce? They do so by asexual reproduction. Okay, especially eukaryotic organism or unicellular organism. So they grow, and once they increase in size enough, they divide into two. And each daughter cell will be like a new organism. That one will continue its own existence until it equally undergoes asexual reproduction. So asexual reproduction is also a form of mitosis. So that concludes mitosis, right? Let's quickly talk about meiosis, which is the other form of cell division. Specialized type of cell division. There are some features of meiosis that makes it unique. So you can see from the beginning, a specialized type of cell division. So it's not just a division like mitosis. It's a specialized type. And it reduces the chromosome number by half. In mitosis, when the parent has 4 chromosomes, each daughter cell will equal number is reduced by half. If the parent has 4, the daughter cell produced will have 2 to have been reduced. And in its own case, instead of creating 2 daughter cells, it creates 4 daughter cells. Okay, 4 daughter cells. And remember, the 2 daughter cells produced by mitosis are what? They are diploid. But for meiosis, the 4 daughter cells produced are haploid. Okay, so in mitosis, N 4. Okay, in meiosis, N equals 2 because it has been reduced by half. So the 2n will become n, and the 4 will become 2. So n equals. And again, each of the 4 daughter cells produced are genetically distinct from the parent cell that gave rise to them. They don't have the identical genetic content with their parent. So they are different. They fall up from the parent cell. Okay? The process occurs in all sexually reproducing, single cell and multicellular eukaryotes. Any organism that undergoes sexual reproduction, okay, will do so by meiosis. So plants and fungi or dendritic organisms. And not only that, meiosis in its occurrence, okay, gives rise to the production of gametes. Gametes. These gametes are the structure that takes part in reproduction. Sexual reproduction occurs in eukaryotes, and during this sexual reproduction, there is formation of gametes, I've said, which are the which are the sex cells. The gametes are the sex cells. So meiosis leads to the formation of male gametes and female gametes. In higher animals, okay, the male gametes are the spermatozoon, and the female gametes are the ova of the egg. Okay, so when mitosis occurs leading to male gametes, the male gametes is having half of the mass in the somatic cells or that same organism. So the male gamete will contain half of the chromosome. The female gamete will equally contain half of the chromosome. Also, how do we now have the restoration of 2n in the individual? It is when the male gamete fertilizes the female gamete, when they fusion. So you have the restoration. When you had n plus n, we have our 2n. So giving us the diploid number. In meiosis, there are 2 successive divisions. We have Meiosis I and we have Meiosis II. Meiosis I is purely reduction in nature because it uses the chromosome by half. And Meiosis II is also purely division. So Meiosis I is reduction, Meiosis II is division. So why is Meiosis II division? Of course, we can understand that when chromosome is reduced by half, well, that's reduction. So why is Meiosis I division and not multiplication, because we end up having four daughter cells from one single cell. That should be multiplication. But no, it is a division. You know, it is one cell that you divide into four. You don't multiply one cell to get four. So don't look at it as when one becomes four, that should multiply. No, it's not multiplying. It's you divide something. When you have a whole, when you have one apple, when you cut it, do you now have two apples? No, because you divided it into two halves, So that's why Meiosis II is said to be division. And I've said it produces four haploid cells, okay? And when you now combine Meiosis I and Meiosis II, you can refer to it as a reduction division process. So at the same time, it's reducing, and at that same time, is dividing. So it's a reduction division. So Meiosis I is reduction, Meiosis II is division. So we get to see how the chromosome becomes reduced and how the division equally takes place. The reduction division, which is meiosis, occur in the formation of gametes. I've said that both the male and female. The male gametes are the sperm or the spermatozoa. Spermatozoa is the correct name. Sperm is just a shortened form of it. Shortened form of spermatozoa. But that does not mean when you call it sperm cell, that's wrong. It's still okay, but it's a shortened form of it. Then the female sex cell or the hello, there was a network issue. So I think it's back. Can you hear me now?
Yes,
Yes,
sir. We
sir.
can hear you, sir.
Yes, sir.
Okay, but there was a time the
Yes, sir.
network went off, right?
Yes, sir.
Okay. Those on the YouTube, are they still on? I want somebody to tell me so that if there's a need for me to resend the link, because the Zoom went off, did it not affect the YouTube?
Yes, it's still on, sir. It's still connected.
Oh, okay. So let me reshare the slides.
No, the YouTube is still on. It's still connected.
Eh, but on the Zoom it's not there again. Have you seen it on the Zoom now?
Yes, sir.
Sir.
Okay.
Yes, sir. We have seen it, sir.
Yes, sir.
We
We
can
can
see it.
Okay. All right. So this is where we were before the thing went off. So meiosis occurs in the formation of gametes. As said, the male gametes, which are the sperm or spermatozoa, and the female gametes, which are the ova. That's in animal. But in plants, especially higher plants, the male gametes are the microspore, and the female gametes are the megaspores. So, however, meiosis is a one-way process. So you can see meiosis being a one-way process as against mitosis, which engage in a cell cycle. So meiosis cannot be said to engage in a cell cycle. That means when a cell undergoes meiosis, it terminates at the formation of gametes. The gametes cannot go again into another cycle. No, because they are reduced already. They are reduced by half. So it's a one-way. Once the formation of gametes is complete, then it stops. Okay? So because of that, meiosis cannot be said to engage in a cell cycle as mitosis does. However, the preparatory steps that lead up to meiosis are identical in pattern and named to the interphase of the mitotic cell cycle. You know, when a cell, okay, is about dividing, for instance, mitosis, I told you it, we pass through some stages. We have the G1, we have the S, and we have the G2, okay, before I now go into M phase, which is the mitosis, right? So in mitosis, once it enters into mitosis and complete mitosis, it comes back into the cycle, and the cycle continues. But in meiosis, it's a one-way process. It cannot undergo or engage in a cell cycle. But a cell that wants to undergo meiosis will equally pass through the preparatory steps leading to actual meiosis. So that means it will equally pass through G1, S phase, and G2. Okay, then enter into meiosis. But the product of meiosis cannot go back and start the cycle again. So that's what I want to bring out there. So it's a one-way process and cannot engage in cell cycle. In meiosis as well, there are two stages, which I have said, meiosis I and meiosis II. I said meiosis I is reduction, and meiosis II is what? Is division. And again, each of these stages is subdivided into 4 phases. So meiosis I is divided into 4 phases, and meiosis II is also divided into 4 phases. But how do you now know a phase in meiosis I as against a phase in meiosis II? Take note of this. For a complete meiosis, we must have meiosis I and meiosis II. So it doesn't mean meiosis I, okay, is a separate meiosis, and meiosis II is another separate meiosis. No, they are together. For a complete meiotic stage, we must have meiosis I and meiosis II. So once meiosis I is complete, the product quickly undergoes meiosis II, or maybe paused after meiosis I before meiosis II. Okay, if there is a pause after meiosis I, then meiosis is not complete yet until it resumes to complete meiosis II to now have the product. So let's see the 4 sub stages in each of the meiosis I and II. For meiosis I, okay, I said, how do you now distinguish a phase in meiosis I from a phase in meiosis II? It's very simple. The two of them have four stages: prophase, metaphase, anaphase, and telophase. So when you are talking of meiosis I, you put the number in front to indicate that this is a stage in meiosis I. So when you see prophase I, that means it is prophase I of meiosis I. So also metaphase I, anaphase I, and telophase II. Under meiosis I, you can never see metaphase II. You can only see metaphase I. Metaphase II will occur under meiosis II. So let's see what happened in meiosis I. Then we equally have meiosis II as well, distinguished by putting the number in front: prophase II, metaphase II, anaphase II, and telophase II. Okay, but much of the process of meiosis II is similar to mitosis. Much of what happens at meiosis II is similar to mitosis. Let's get there. Now, to start with, the phases of meiosis. Starting with meiosis I, okay, what are the main occurrences in each stage? For meiosis prophase I of meiosis I, there is synapses and crossing over. Okay, the chromosome inside the cell, they pair up. That's synapses. They identify their the one that is similar to them. The pairing up will be among similar chromosomes. If they are not similar, they will never pair up. They can only pair up when they are identical. So, and that is synapses. Synapses means pairing, coming together. Then there is also another phenomenon called crossing over. Crossing over between chromatids of paired chromosomes. You know, each chromosome has 2 chromatids. So when they pair up, there's tendency for chromatid of one chromosome to cross over the chromatids of another chromosome. Then at prophase I, we equally see right. So that's prophase I. In this case, you can have early prophase and late prophase, but the major thing is what happens during prophase I. We have synapses coming together, crossing over, okay, chromosome condensed. You will observe that these are equally the same event that occur under prophase of mitosis. Prophase of mitosis. The difference here is pairing up. In prophase of mitosis, Each chromosome behaves individually, but here they behave as a pair. They first of all identify each other, and those that are identical, they pair up. All of them, they pair up. They occur in pairs. Then the chromosome become visible as well. Then spindle fibers form, nuclear envelope disintegrate, disappear. Okay, then the spindle fiber that appears are attached to chromosome, the kinetochores on each chromosome. They are attached to the kinetochore of each chromosome. Then going on to metaphase 1, the pair of chromosomes align along the equator. They still occur in pairs. So you now have the pairs aligning up. Unlike in prophase, unlike in metaphase, where the individual chromosomes will align. Here it is the pair, each pair. Each pair contains two chromosomes and 4 chromatids. Don't forget to. So they now pair up and align at the equator. Then at anaphase 1. So at prophase, at metaphase, the occurrence there is the alignment of chromosomes. But what are the nature of the chromosome? In prophase, the chromosome is single. Each of them will align up singly. But in metaphase 1 of meiosis, they just don't align singly. They first of all pair up, and then it is the pair. So each pair will now align at the equator. I'm trying to bring the screen. Let's see the screen.
Yes, sir.
Yes, sir. Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. I
Yes,
hear
sir.
you.
Yes, sir.
Yes, sir.
Yes, sir.
Are you guys drawn? What is the meaning of all that?
Sir, you are no longer sharing your screen.
I'm not seeing the screen again. What are you trying to say?
Yes, sir. We can see.
Yes, sir.
Yes, sir. We can see it.
Yes, sir. We can see.
There is no
Yes, sir.
slide again, sir.
Sir, we can't see any slide.
You will see now. Are you seeing now?
You
Can
still
you
loading?
see? Can you see?
Yeah. Yes,
You
you
can
can see
Yes,
now.
sir.
Yes, sir.
We can see it now, sir.
Yes,
Yes,
sir.
sir.
Okay. So the slide generally summarizes all the major events in each of Meiosis I, Phase I, Metaphase I, then Anaphase I, and Telophase I. So now, when you look at, because it is in this Meiosis I that we have, you need to listen attentively and be very, very careful. Like I said, Meiosis II is just like mitosis, which you have talked about. So, but what makes Meiosis I very, very unique is especially Prophase I, where you have crossing over, leading to exchange of some portion of the chromatids. So it is at this Prophase I that you have gametes, okay, that will not contain the same arrangement of genetic information as you have in their parents, because there has been crossing over, alignment and realignment. So this is the basis of variation that is being noticed in an individual. When you look at an individual and the parents, as much as they may be identical, they can never be 100% identical. There will be that variation, distinguishing one from the other. Okay, that is between offspring and parent. But when you now look offspring of the same parent, they still have that distinguishing features on each of them. It is because of the genetic crossing over that has taken place, while the gametes that fertilizes the egg is being produced. So it takes place in both male and female, not just in one. Who is that? Tusaya Dane, what is happening? Better mute yourself. So, and when you now go to, then another thing is the pairing of chromosomes, so that they now have what is called tetrad. Tetrad means four. For what? 4 chromatids. When you see them pairing up, the pairing up is so close that you will now think it's just a chromosome that contains 4 chromatids. But a chromosome cannot contain 4 chromatids. So that is 2 chromosomes that looks like one, and they have 4 chromatids. So they are now called tetrads. In that form, they are called tetrad. So this is what leads to the formation of variation. Now, the 2 members of a pair, they are said to be homologous. So if they are not homologous, they cannot pair up. They will only pair up with their own homologue. They will only pair with their own homologue. And by doing so, they can now exchange portion or parts of their chromatids. So a chromosome contains 2 chromatids. So those 2 chromatids are called sister chromatids. They are in the same chromosome. But when they pair up, you can now have crossing over between non-sister chromatids. That is, chromatids of a chromosome will cross over the chromatids of another chromosome. The 2 chromatids in the chromosome cannot cross over each other. Sister chromatids do not cross over each other. Crossing over occurs between non-sister chromatids. So that's why I'm emphasizing all the occurrences in prophase, all the events that take place in prophase, because That is where the major event takes place. Now, when you move on to metaphase, you will see that they now align at the equator, but it's a pair. The two chromosomes that pair up, that's homologous chromosome. We call them homologous chromosome. They pair up and they still align like that at at metaphase stage. They pair up and they align in such pair on the equator. So that is at metaphase 1. Now at anaphase 1, okay, anaphase in mitosis, what happened there is when they align at the equator at metaphase, the spindle fiber will attach to them, and at anaphase, the spindle fiber will start shortening, and by the time they are shortening, they move the chromosome with which they attach to, they move it towards the pole. Now, a pair align at the, now have the spindle fiber attaching to them. So at anaphase, the pair will now be separated, not the chromatids now. It is the homologous chromosome that pair up, they will now be separated by the movement of the spindle fiber towards their respective poles. So you now have a complete chromosome moving up and another complete chromosome moving down from that pairing. The pair now becomes separated so that you now have separate chromosome. One moves up, the other moves down. What happened to a pair will also happen to other pairs of homologous chromosomes that pair together. Like you can see in this diagram, you see 2 green and 2 blue. So they pair up at anaphase, and at telophase, they have gotten to their respective pole. And like we said, what happens at prophase is the opposite of what happened at the telophase. So at telophase, the chromosome is now being surrounded by nuclear membrane, because nuclear membrane will appear again at telophase. It will appear. So at telophase, nuclear envelopes partially assemble round form around them of mitosis. So the same will equally happen here, but what we now have at the opposite pole will now be a complete member of each pair. Complete member of each pair. So the spindle fiber that form at prophase will disappear at telophase. So also occurrence of prophase at the opposite of what happened during telophase. So the prophase 1 that I said is a critical stage is further broken down to reveal the substage in that broken one. Okay, We have the leptotin, we have the zygotin, quercetin, diplotin, and diakinesis. So, so all this with their unique the first stage of Prophase I, where the chromosome starts to condense, because the chromosome needs to condense. Prophase, whether mitosis or meiosis, has to do with condensation of chromosome to make it more visible. Still in Prophase I, zygotin, that's synapses. After they condense, they can now recognize each other as being homologous. So the homologous chromosome will pair. So they pair up to form pairs of chromosomes. Each one of them is now called bivalent. Bivalent, consisting of four consisting of 2 chromosomes but 4 chromatids. So bivalent, bi means 2. So bivalent, that's 2 chromosomes. Okay, we can also refer to them as 4 chromatids, which is tetrons. When you look at that, when you look at a pair, you see 4 tetrons there. Quercetin. Quercetin, this is the Prophase I stage where there is a crossing over. However, the crossing over will be between sister chromatids, non-sister chromatids of homologous chromosome. It can never be crossing between sister chromatids. No, it is between non-sister chromatids. So a chromatid from a chromosome will cross over the chromatids of the other chromosome, though they occur in pair, 2 chromosomes pairing up. At diplotin, homologous chromosomes start to separate but remain attached by chiasmata. You know, the point at which non-sister chromatids cross over each other is called chiasma, singular. Chiasmata singular, plural. Is that okay? At diakinesis, homologous chromosomes start to separate. Okay, diplotin, homologous chromosomes start to separate, but remain attached by chiasmata. So after the crossing over, they will have exchanged some portion of their genetic makeup. So they go into diplotin like that to separate. But when they are separating, they are no longer the way they were. They have exchanged something. So when they are now separating now, it cannot be the same chromosome before undergoing that's crossing over. So they separate at diplotin, but the point at where they are still attached, that's the chiasmata, they are still held. So diakinesis, chromosome continue to separate, and chiasmata move to the ends of the chromosome, separate to now give their respective individual chromosomes. Okay, individual chromosome will now be on their own. And this is what we go on to Prophase I, to Metaphase I, Anaphase I, and Telophase I. So at Telophase I, of course, the pair anaphase, they will separate. So you have single or individual chromosome moving to a response. So single chromosome now, not chromatids, unlike in mitosis, where a chromosome will separate and you have two chromatids, each moving to respective opposite. Here, they will still be together, okay, until they get to anaphase, where they will be separated. And you now have one member in one chromosome, in one cell, and the other member in another cell. Okay. So this slide is illustrating how Prophase I takes place with special reference to crossing over. And let me show you so look at leptotin. The chromosome are separated, but they are now moving together. So azygotin, they are together. See, at pachitin, you see them, they have exchanged some portion. So this one will clearly show it. This is when they are still individual, when they are still separate, okay, then they now pair up, okay, pairing up, that's a good thing. Then at pachitin, you see them exchanging portion. So when you look at this blue is a complete chromosome. This red is another complete chromosome. So look at the red and the blue. Towards this side, you see the red chromatid crossing over the blue chromatids. And at the end, you now have them separated, but they are not like what they were before. One is a complete blue, the other is a complete red. But now you have blue with some touch of red, and you have the red with some touch of blue. So they have exchanged some portions of their chromatids at this length. That's where they extend. So this one, okay, when you look at each chromatid in each of them, there is a complete chromatid and there is one that is not. Then when you look at the other one, there is a complete red chromatid and there is a red with little touch of red, of blue. So when you now have that, the one that is still completely blue is said to be non-recombinant. And the one that is with a touch of the other color are called recombinant chromatids. Recombinant, because they have taken something and incorporated it into themselves. So they are recombinant. The one where you have the touch of the other one, you have the recombinant. And then the red, the touch of blue, is also a recombinant. So this is another one showing crossing over. Okay, the blue and red chromosome giving us recombinants. Anywhere you see chromosome with a touch of red. So these are other illustrations of crossing over when they are labeled to show which part has crossed to the other part. So for the blue, we have capital A, B, C. For the red, we have small letter A, B, C. But they cross over each other. Blue crossing over red, and then the bell. Okay, and then the other one, which is the red, has small letter A, B, C. So after the crossing over, you can see there is a complete blue, but the other one that crossed over blue, okay, has altered the arrangement of the number. So for instance, instead of having A, B, C, okay, for this one that is not involved, we now see this one now is having complete blue, then red plus red plus some portion of blue on it. So you now see that instead of capital A, B, C, we now have capital A, capital B, and small c. So small c has come from the red. And let's see what happened to the red. Instead of having small letter A, B, C, you now see that one has crossed there. So that's for crossing over. Then I've explained metaphase after prophase, metaphase, anaphase, and what really happened. So this slide is showing Meiosis I and II compare when you want to take the two side by side. So Meiosis I is what I just explained, that it has so many things that mitosis does not have. And by the side, you see Meiosis II. Meiosis II, which we said is purely mitosis in nature. Meiosis II is purely mitosis in nature. Take note. So as a form of comparison between meiosis and meiosis, we look at the comparison. We said meiosis, the one on the left side is meiosis. The one on the right side is the mitosis. So the two start with one cell each, one cell each. So while in meiosis, the chromosome, the pair up. In mitosis, you can see they are wandering. Okay, just moving, they are not pairing. Each of the chromosomes is just moving freely. But in meiosis, at prophase, they pair up. Uluoma, will you shut down your video? Is there anyone who is interested in seeing you on the bed? What kind of nonsense is that? So this slide is illustrating in pictorial form what I have told you earlier concerning differences, though they all started with cell, but when you look at meiosis, it undergoes two stages, Meiosis I and Meiosis II, leading to 4 daughter cells. For mitosis, is just one, and at meiosis, okay, you see it dividing into 2, and each one continues this and this. Now let's quickly look at the importance, the significance of meiosis. In the production of gametes, I said that meiosis enables the production of gametes. So the gametes that are produced when they fertilize each other, okay, we now have a restoration of the diploid number. So I said it reduces it by half. So the gamete has only half of the number of chromosomes as in the parent cell. And this is very necessary because if there is no reduction in the cell, that means if it fertilizes, if a male gamete fertilizes a female gamete, and both of them has 2 eggs, so the resulting zygote will be 4N, which is a super something. So it has to be reduced so that after fertilization, the number can be restored. So by doing so, meiosis facilitates stable sexual reproduction. So also, maintenance of chromosome number in somatic cells. Somatic cell, for instance, of a normal human being contains 46. So, and that 46 has to be maintained from generation to generation. So it means for an individual to have that 46, the gametes produced each of them must have half, so that when they meet again, you will now have half plus half to have our 1. That is 22 plus 22 to have 25. 46 has to be divided into 2 equal parts. That's 23. So in a normal human being, this gametes will contain 23. Each gamete, whether male gamete or female gamete, will contain 23. So that when there is fertilization between male gamete and female gamete, you can have a restoration of 46. Then, this is the production of variation. This I explained while I was talking about Prophase I. Because Prophase I, where you have exchange of some parts of the non-sister chromatids. So that makes them to be unique, to vary from what we have from the parents. So new individuals do not resemble their parents closely. When you look at new individuals, they don't resemble their parents closely, okay? And this is brought about in 2 ways. First, there is a different combination of chromosomes due to reshufflement of parental chromosome during meiotic process. Okay, different combination of chromosomes due to reshufflement of parental chromosomes during meiotic process. And 2, crossing over 2, okay, brings about variation. When chromosomes are arranged along the equatorial plane, there is crossing over. So in the process, the chromatids exchange genes and so increase genetic variation, okay, which enable the species to survive in many environments. So these are importance of meiosis as well. So any question? This is where we are going to call it a day. By God's grace, we meet on Friday, same time. Hope there will be no each so that we can start on time by our scheduled time. So any question? I'm waiting for your question now. If you have any question, just raise up your hand. Joseph Hilary. Joseph Hilary, can you ask your question? Unmute and talk. Joseph Hilary, can't hear you. Okay, let's have Ajayi say enough.
Yes,
Ajayi
sir.
say
Good morning,
enough.
sir.
Can you ask your question?
Yes, sir. Good morning, sir.
Morning. Is that what?
Sir, is pre-metaphase part of the stages for mitosis?
Is what?
Hello. I said, is pre-metaphase part of the stages for mitosis, or just between prophase and metaphase?
Okay, pre-metaphase. No.
Yes.
4 stages. You know, in mitosis as well, it could have been 4, but there are some textbooks that included prometaphysis. And
Yes,
when
sir.
you look at prometaphysis, it's just something that starts from prophase, okay, and continues in
Yes,
that
sir.
stage, and something that will equally continue in metaphase. That's prometaphysis.
Yes, sir.
So in
Yes, sir.
meiosis, it's not.
Okay, sir.
Iberedem Emma. Iberedem Emma. Unmute yourself and ask your question. Are you not there again? I can't hear you. Hilary, I can't hear you. Maybe your question has been answered by the one I told the last speaker.
Good morning, sir. Good morning, sir.
Iberedem.
Yes, sir.
Good morning.
Good morning,
Who is
sir.
this?
It's Iberedem,
Okay,
Emma.
are you Iberedem?
Yes,
Okay.
sir. So can interface be taken as a stage? Because some people say it's the resting stage that is not, cannot be taken at the stage of cell division. Can it?
You joined this class late. You joined this class very, very late. I said it when I was there. I said interface is never part of mitosis. Interface is a stage, okay, where activities that will lead to mitosis take place. So it's not a resting stage. It's a very active stage, not rest, you know. Now,
Okay,
let me
sir.
ask you a question to know
Okay,
that interface is not a resting stage. Okay? Now,
yes, sir.
you are here in school studying to achieve an aim, right?
Yes, sir.
This particular course, before you pass it, you must write an exam, right?
Yes, sir. Yes, sir.
Yes, you must write an exam, you must pass it. Okay? Is
Yes,
the
sir.
exam tomorrow?
No, sir.
The exam is tomorrow.
No, sir. No, sir.
When is the exam?
Around March.
I know. When do you know when the exam will come up?
March.
Okay,
Yes, sir.
around March. And now, don't worry. And now you are in class, trying to learn so that you can pass your
Yes,
exam.
sir.
The essence of you being in class is to learn so that when the exam comes, you can pass.
Yes, sir.
So the same thing happens to interface. Interface is like you are nodding. The division has not come up, but you will need your study now for the exam. So interface is like preparing all the material, the needed thing for cell division, for mitosis to take place. So you don't just wake up and say, cell with this. What will you need? The same way you will not go and sleep and just come on the day of the exam. You know? So
yes, sir.
are you now resting?
No, sir, I'm not resting, sir.
You are resting.
No, I'm not resting. Oh.
I'm preparing. You are attending classes, you are studying.
Yes, sir. Yes, sir. Yes, sir.
So that is the same thing for interface. Will you now say interface is a resting phase? No, it's not.
Yes, sir. Thank you, sir. I've understood you, sir. Thank you, sir.
Any other one? Yeah. Another one? No more question. You better mute yourself.
Okay, sir.
Yourself. So, if there's no more question, who is that? Who want to talk? Anybody want to talk? So, in the case of any other question, we round up for today. We meet again on Friday by God's grace. And hopefully there will be no technical hitch and we can start our class on time. Enjoy the rest of your day. Thank you. And God
Thank you,
bless.
sir.